in the middle of a lot of discussion for the course. We also want to include a little bit about some of the genetics and ethics of if you're thinking about monitoring molecules, monitoring spatial data. At some point, there's going to be a question of what else we'd measure, what else you could make. So there's a bit of an ethics discussion, and also some of the new technologies for CRISPR that I want to talk about in applications. I'll start with a bit of background. There's actually a similar lecture that the medical students get in the fall. And so this is a bit of a preview of some other lectures, but okay. So I think as many of you probably have thought at some point in your life, is you feel like something, we all get a genetic shuffle at birth. You all started out as a single-celled embryo. I've already told you about what I remember when I was an embryo. Hopefully you have forgotten that, but based on what your faces say, some still remember that. It's recorded as well, so it'll always be online, I guess, in some dark channel of the internet. But in case, when you're an embryo, when you get your genetic shuffle as an embryo, sometimes you could say that you win the genetic lottery, and I'll come back, put it in quotes for a reason. I'll come back to that later. But some really fun examples are, for example, when you get mutations in myostatin, you can have really huge muscles even as a child. If you wonder what the ortholog looks like in animals, it can look like this. You can have these really giant muscular animals, and this is something you can think about specific mutations and specific genes, or a lack of that gene. In this case, gives you really interesting phenotype. Other examples are LRP5 mutations, for example, can lead to really strong bones. This was kind of the concept behind an idea called unbreakable. Excuse me. Has anyone seen this movie, or anyone? A few people, but I generally stop using cultural references in my lectures because I'm getting too old. But this is one that's it was a real movie that was about a guy with unbreakable bones. And it was based on actually an idea that you could have bones that are so dense that the original case report for LRP5 was a student, actually by Rick Lifton, who's now at Rockefeller. He's the head of Rockefeller. He was on that original paper that had trouble swimming because his bones were so dense, he would start to sink every time. So it's like an interesting case report where it's a good example. We're always look for odd people because they might have an interesting story, genetically or otherwise, to talk about. And, it's not quite like a superhero like Spider-Man, but it gets you as close as you can get. There's also examples, again, found by case reports, what was called the Berlin patient, which I'll show in a second, was you can have a certain mutation that can prevent the HIV virus from getting into your T cells if you have a mutation in a receptor, like CCR5, for example, you can see here. So if you have this mutation, which was first found, you can see here back in 2009, was a bone marrow transplant, a stem cell transplant to help an HIV positive patient. But then after a while, I realized you didn't really need to, the counts were staying low even off the retroviral therapies, which is extraordinary. So if you have a specific deletion, you're called CCR5 delta 32, you can have this, immunity to HIV, almost all strains of HIV, kind of like a genetic superpower. Not quite a full superpower, but it gets you pretty close. And there's another good example. So those are cases where you can win a superpower field, but also there's some cases where you can have a high risk factor. And even some of the discussions we just had are about protein interactions or risk alleles. In some cases, it can lead to losing the genetic lottery. For example, there's mutations in genes for hypertrophic cardiomyopathy, which is actually the most common cause of sudden death in young athletes. This can lead to outflow obstruction, lead to a really early heart attack. And so Reggie Lewis sadly died of this. There's other athletes who have died of a sudden heart attack at the prime, in their mid-20s, let's say, in great shape, and suddenly dying quickly. And several of them have been found to have mutations in genes related to hypertrophic cardiomyopathy. And so this is, gives you a risk factor. And there's actually, so that's kind of the background. You have genes that are risk alleles, genes that are protective alleles, well, variations of good and bad genes, if you will. But an example where things went wrong, though, in this sense, ethically, is that Eddie Curry, for example, and this is now in the mid-noughties. Remember the mid-noughties? Anyone from the first lecture? The mid-noughties. In the mid-noughties, was that he had, he was doing well in the season. He was 22 years old. He was doing great, but he had hospitalized with an irregular heartbeat, and he missed the last 13 games of the season. And then heart specialists had cleared him, say, okay, seems like he's okay to resume practice. But then at the time, about 20 years ago, so the human genome first draft was finished in 2000. First update was really in 2003. So the genomics world was starting to percolate into the society. And the bullish general manager said, I understand there's privacy issues, but I really want him to get tested for whether he's got risk factors for these genes, because he doesn't want to have another Reggie Lewis situation or Hank Gathers, basically places where players died right on the court when they had some of these risk factors. And so Eddie Curry considered this offer, and they actually offered him an annuity of $400,000 per year for 50 years if he took and failed the test, meaning he had the risk alleles. And this is a pretty good offer, right? I feel like I don't know whatever test I'd be taking. I feel like I would just say yes to that. It's a pretty good deal. But he refused, but more unethical grounds. He said, it's my DNA. You shouldn't discriminate me based on something I had no choice on. It's just the genes I may or may not carry. So he refused to take the test, and then later was actually traded to the Knicks, then to the Timberwolves, then the Heat, and the Mavericks, and the Golden Bulls, and eventually the Zhuhai Warriors in China, and has kind of been kicked around the NBA and other sports groups for the last 15, 20 years. And we still don't know whether he had risk alleles, but we just know that there is some pressure from the basically some employers that I want to know what your risk alleles are. I want to know if you're we're investing money and time and energy into you, and I don't care if there's privacy issues. I just want to know. But I think to his credit, he refused, and I don't know exactly where he is now or how much money he has, but I think the bravery there was pretty striking, given also the offer. And so this I think indicates there's other examples like at Burlington, the Santa Fe Railroad Company. They were testing their employees for carpal tunnel syndrome. I'll show you a few other examples where this has been deployed. But maybe a change was needed, as was discussed in the mid noughties, and in the 2008, George Bush signed HR 493, which is the Genetic Information Non-Discrimination Act. So finally, this did come to a head. It had been discussed actually for 15 years at some point by the time it was signed. The biggest worry was insurance agencies wanted to say, well, but we need these risk models to help give us better premiums and make it so we don't die as an industry. Generally, I feel like you should take whatever insurance agent tells you with a little bit of grain of salt, a little bit of suspicion. I once had renter's insurance in an apartment in Brooklyn, and then like the cabinets fell down once, and then on the phone I said, hey, I have renter's insurance. I'd like to, all of our dishes are broken. I'd like to submit a claim. And they said, well, tell me what you think was wrong. I said, well, I don't know. I didn't build the apartment, but it looks like they missed all the studs when they installed the cabinets in our kitchen. And the insurance agent just said, oh, that sounds like bad construction. We don't cover bad construction. So it's just a lesson I can tell you. If you're ever filing a claim, thinking about insurance, just say, I don't know what happened, but it's bad and I need it replaced. And do you know why it happened? I don't know why it happened. What happened? No, just know that I no longer have cabinets on my walls and I'd like to file a claim. Anyway, the insurance companies will always find a way to say, well, but we want to use this part of our model or look for a way out of it, is my point of that personal anecdote. But it was signed into law. And particularly, I want to highlight what it protects against is that it protects against people being discriminated against for health insurance. So you can't lose your eligibility, your coverage, or underwriting, or different premiums. But before this was signed, someone could say, oh, I have a BRCA1 mutation for risk for breast cancer, for example. The insurance company could have said, well, we're going to give you a higher rate. In fact, my then girlfriend, now wife, had this problem before this was signed, is that she didn't want to get any insurance because she had BRCA mutations and really was terrified to talk to any insurance agent. It would have been legal for them to ask her, give us your genome, or we're going to do a genetic test and then give you higher rates. So until this was signed, this is something she was personally very afraid of. And so we're glad when this happened. But then also for employment. So you can't tell us, for hiring, firing, promotions, pay. Someone can say, I don't like your genome. I'm not going to give you a promotion. I don't know who would say that, but people, again, will often find weird ways to not give you promotions, sometimes for reasons of, racism or other problems. But this is now illegal. So you can't say, I'm not going to hire, fire you, or change your job based on, or, other covered entities from acquiring or requesting this genetic information. So Geno's protecting there. But it's interesting. There are some things it does not cover, which is an interesting little legislative background, is that if you have 15 or fewer employees, it does not cover you. Mostly, you might be wondering, like, why would a small business not need to follow the law? In this case, the idea was that it's too onerous for a small business to have to accommodate these changes. So weirdly, and this is common, it turns out, amongst a lot of laws. If you're a really small business, you don't have a whole legal team. You just have maybe 10 people. You're not going to have time or energy to really check every aspect of this. And so it doesn't mean you shouldn't do it. You can still have other problems. I'll show you in a second, but it does. There's one carve out. Also, the military is not covered by it. And long-term care insurance or life insurance or disability insurance. You might think, what if I have ApoE risk alleles for Alzheimer's and you want to get long-term care possible insurance? A company could technically ask you, well, we want to see what your risk factors are. I want to see your 23andMe report. But I'll come back to that, not yet in Florida. Speaking of, so that was true everywhere except in Florida as of 2020. They were the first state to say, let's protect you from life and disability insurers, and that make it so. Again, the concept of the ethic behind it is you get a shuffle of genes. It's not your choice what you got. So how can you be penalized for something that you had no choice in? Right. So it's not like you're like driving fast, and younger people have a higher rate because they get in more accidents. They generally do more stupid things when they're younger. I think we all do more stupid things when you're younger, but there's something you can choose that you're affecting how you drive. In the case of genetics, you obviously don't have that choice, at least mostly, and I'll come back to this at the end. But Florida, to its credit, really expanded this protection, these genetic protections, to make sure you can't be discriminated against based, again, on your genome. for long-term care and disability insurance. Actually, have if you're someone in your family has any given disease, cancer, it could be ALS, it could be Alzheimer's, you can't be discriminated against if you want to start to prepare just in case if you think you're at risk. And again, the insurance companies want to say, well, we want to know that risk because we want to adjust our claims and proportions, but they so far can't do it in Florida. Okay, so when has this law been used? So that's a bit of background about genetics and privacy and some of the ethics, and I'll come back to some other applications in the lecture. But the first real test of the 2008 law was in 2015. Was a great case called the Mystery of the Devious Defecator. Any of you ever heard of this case? It's one of my favorite case reports of interesting litigation and genetic discrimination. So there was something called the Atlas Logistics Group that had warehouses for storing products, sold to grocery stores. And there was a mystery employee that began habitually defecating in one of its warehouses. And so you can imagine they were not happy about this because, well, I mean, no one would be happy about habitual defecation in a place where you'd like to keep it clean and organized, right? So this is an interesting case that was filed. And Atlas Logistics, they got in trouble because the managers wanted to know who it was. They're like, well, who's doing this? And so it's great, of course, from stools, you can get microbial DNA, but also human DNA. So they said, well, let's actually go and test. We want to test the stool, and we want to ask all the other employees who we think it is for cheek swabs to see, well, we think it's you, the one leaving it behind. So you think, well, this is, it's left behind. Is it really a privacy consideration? It's just left on the floor. Who's responsible? But again, you're not supposed to ask people for their genetic information, but they did. And the fun little note about this, apparently it's not as rare as you might imagine. There's a whole another article about other employees who have been defecating in hallways. Fun fact, if you want to read about it, there's another article about it. But in any case, this was the first big test of the law. And these two gentlemen were asked to provide DNA tests and samples. And they said, it's not us. We're not going to give you our samples. It's not, you have no right to ask us. And someone gladly informed them that it's their rights to reject that request. And it was the big test of it. And then they did actually win. It wasn't even them. So they weren't the ones doing the mysterious defecation. But what's great about this is it was, essentially a 2.25 million, claim or decision. And some of us also punitive to really, make sure that people don't do this again in the future. So the first big test of the law is the way to kind of make sure you can keep your privacy. So this was a good article in Nature about it, which was also kind of fascinating mental experiment. And if you think you want to do this to your employer, I would encourage you not to, just because you do still leave human DNA behind. I'll get back into that. Actually, not now. So all the DNA you leave behind tells a story. Again, it's in stool. There's human and microbial DNA. And you are an echo of your ancestors. So we left Africa approximately 100,000 years ago. And this is the broad general scope of where we think humans have migrated across our planet over the past 100,000 years. And because of that, we all have variations of some genetic similarities, but also some differences that due to population bottlenecks that define hair color, eye color, skin color, features, different facts about people's ancestry. And this was something that was actually put into a really famous paper back in 2008 of just taking genetics data, genotyping data, and say, can we remake the map of Europe just by genotyping data alone? And you can see it's not perfect, but it actually does a pretty good job of just separating out. You just take genetics data, how close can you get, and see how close they are in sort of this principal component analysis map. Now, PCA plots are not supposed to normally give you distance as a function of a real value, but here it actually looked quite similar to the actual map of Europe. And you can see, what's the genetic correlation as a function of distance. The closer you are to where your ancestors are, you have a little bit more of a correlation, and then it starts to dissipate as you get thousands of kilometers away. But they did also claim that they could get to within a few hundred kilometers where you're likely born based on just your genetics alone, mostly on Europe. But it shows that, it's not perfect, but if you can take just a cheek swab, I can tell you where you're possibly likely from within a few hundred kilometers with a certain error bar. It's not perfect, but it does show you it's possible. Now, we've actually used some of these same methods in other papers. So, for example, we're inspired by Heather Dewey Harburg. She would take cigarette butts left in New York City, take the cigarette butt and actually run genotyping arrays and then see what the person likely looked like based on hair, ancestry prediction, and make these kind of creepy looking maps of what we think the person looked like. And it wasn't perfect, but it was kind of a fun artistic rendering of, I know who left that cigarette butt, I will find you in your face like this. Which is kind of a fascinating example of taking public genotyping data to make a face. We are inspired by this, actually published this about 10 years ago to say, in the subway system, could you reconstruct the United States Census data based on the human DNA left behind? And can you see this molecular echo of who's there? And again, it's not perfect. No prediction method on ancestry is, but you can see those, Chinatown look more Asian. Yes. Does Spanish Harlem look more Colombian, Mexican, Puerto Rican? Yes. And then does Canarsie look more African American? Yes, actually. So it's not, again, not perfect, but you can see these different areas of New York City even carry this echo of who lives there in that area, which is a fascinating example, in my opinion. But you're also an echo of your family. So keep in mind, it's not just your echo of your ancestors, but this is the rate of how similar you are to your cousins, your second cousins. It's a fun thing to do at a family gathering. When you see your cousin next time, you say, "Oh, we share 12.5% of our DNA." It's always a fun, really dorky genetics fact that will immediately label you as a dorky scientist, but I still think it's a fascinating number to throw around at a family gathering. And so because of this, you can do what's called familial DNA searching. So even if you haven't committed a crime, you get enough of the FBI markers. Actually, it's these FBI markers that they have these 4 loci and 7 additional ones that can track, based on your genotyping data. Are you in the database? But again, even if you haven't committed that crime, if you have a sibling or a first cousin, some of your markers are already in the FBI database. So you already know you shouldn't commit a crime, but if you have any family members that are already in this database, you should definitely not commit a crime because they find you through these methods. Okay, so that's on the genetics. What can other molecules reveal about you? You just heard a little bit about molecular age or tissue-based age, but if you remember from the epigenetics lecture, this is the same couple slides as a refresher, is that you can predict your age based on epigenetic age. And actually the plots that Andre was just showing actually show a similar degree of variation of this kind of idea of what's your predicted age versus chronological age by tissue, but you can see it here. And we've even used this to actually take blood left at the scene of a crime to predict the age of a person that had left that blood. This is a case for Stephen Avery, which actually let's find out that was fresh blood, not bank blood from this case. So you could use it at crime scenes, for example. This has led to a whole bunch of companies now trying to commercialize some of these ideas of how old are you really in these commercial tests. So there's, for example, I have no relationship with this company. I just think it's interesting that they can do, as you can see here, you can do epigenetic services, predict your biological age. And even if you make iPS cells, they can say, did you really make iPS cells or has your reprogramming not been complete yet? So they can do this kind of validation for you. And they have a paper about this on the right. So interesting applications of let's confirm that you've done the reprogramming you think you have. And I'll come back to this at the end because it's important to think about in general reprogramming options. It also could someday be important for how do you determine whether or not you should get into a bar. You're all older now, but you probably remember when you were younger, it was very hard to get into a bar. You'd maybe get a fake ID from your sibling or someone. These days, you can go to YOLO IDs or sites like and get a fake ID from any state in the union. Yes, you can pay by Bitcoin. So you can imagine in future, well, you'll check age at the bar with like a nanopore sequencer or something. I'd just like to have as a thought experiment. No one else is talking about this, but I just fascinating to think about. And this led to some people to think, well, maybe we should update GINA because there's microbial privacy. I'll show you in a second. There's epigenetics markers. You could try and predict a lot about a person based more than just on DNA. So this has been discussed in the past couple of years in the news. It hasn't happened yet, except for Florida, but it is something just to think about: is what else can reveal privacy. So to that end, I'll give you a few other vignettes about privacy before I get into a story about genetics. And in particular, for example, if you look at the people have published papers looking at, could you take a Kmer signature of microbes in your gut? And if you go to the bathroom in a toilet, you think, oh, I'm just flushing it away, and there it goes. But if someone sequenced every toilet of every part in the city, you could even see where you're going because you all have a unique gut signature, even after fecal micron transplant or antibiotics. And so what's interesting is you all carry a little unique profile in your gut that you could track if you had a sequencer on every toilet in New York City. Now, you would think, who would put a DNA sequencer on every toilet in New York City? Well, I think it'd be fabulous, and it'd be very interesting to see what comes out. We've done it at least for wastewater broadly, but not on every toilet. But this shows there is a way to predict where you are and who you are, which has raised privacy concerns detailed here in this article. For example, that's just at the microbiome scale. Even each shoe has a unique microbiome. So going back to some of the idea of tracking subway studies we published a few years ago, could you predict if you take a random sample, say from a shoe, and what how likely are you to predict what city you came from in the world from the Metasub consortium? We get about 85 to 90% accuracy at the best for predicting your city of origin, which again is not perfect, but it's actually above noise, and so it is possible. So you go to your shoes, and if you this is a privacy paper we published looking at what are all the different layers of omics that could predict things about you, like your genome, your microbiome, your epigenome, other omics layers that could tell things about your forensic or social profile. So this gets into question. The privacy is going to get harder and harder to keep, I would argue, but if there's good protections legally for you, then hopefully that'll be less of an issue. So I think privacy will just get harder to keep because of all these metrics. And so it might make you wonder, could all these methods be legal? Could you patent these methods? Is it legal for someone to just take something that's left behind and sequence it and profile it? Legally, anything you leave on the street is actually fair game. Actually, if you put your garbage on the street and you think the police came and they rummaged through my the garbage, well, first, hopefully you don't have anything that should be in that garbage. But if you do, it is no longer your property once you put it in the garbage. It's considered public domain. And so that's why people can do what's called dumpster diving once it's on the street and then try and find things out about, nefarious actors, companies. No one's done this, yes, based on the look on their faces. Some of you, if you have, you'd be like, oh, I've done that, but no one nodded. So that's probably a good sign. But you might be wondering, is this possible? How possible is this or legal is this? And I point you again to interesting legal case where this was Kylo versus the United States, where he was growing a lot of marijuana in his apartment, as you do. Sometimes people are ambitious and entrepreneurial, and he was selling it. But then the police used a thermographic camera to figure out where the growing apartment was in a building. They narrowed down the building, and he said, "Well, I have a reasonable expectation of privacy that people can't see in the infrared spectrum and see into my house." So he challenged it in court, went all the way to Supreme Court. He won in that case because a reasonable expectation of privacy for the Fourth Amendment doesn't include people having thermographic vision. Now, today that might be harder because it's a lot easier to get this kind of technology, but at the time he won the case. And so a lot of this revolves around legal foundations as well as a reasonable expectation of privacy. And this is where I want to tell you a little vignette of some of my own personal story of, for example, could you patent a gene? This started back in grad school at a bar, as a lot of things do. We're drinking over beers and like, can you patent a gene? I was like, I don't know, could you patent a gene? I was talking to this guy, Don Katz, who was at the law school, and said, we should chat about this more. Is this really possible? I said, I've heard you can patent genes. He said, what do you mean? We should teach a course about this. This led to a special course called Law and Order, like bump, a genomics unit, which we set up at the law school. And what was inspired by this was like, I was a postdoc at the time looking for disease genes, but there could be hundreds of them. And had read this paper late in lab one night. It said that, essentially it estimated that 20% of human genes were patented. That can't be right. That seems really bizarre that they're owned by someone. Because what if I just take a cheek swab and I want to sequence my own gene, which is relatively easy then, it's very easy today, as you've heard about in this course. And so I thought, that just seems really bizarre. How's that possible? And started chatting with other scientists, other researchers, and some of them, for example, were like CCR5 was patented, for example, like this gene we just heard about. And there were people posting online that private companies, own your DNA. And so we actually worked with the ACLU and we filed briefs to challenge it, as well as with patient advocacy groups who say, if you're a breast cancer patient and you just want to get your BRCA1 gene sequenced again, it was impossible for you to do so because you'd be violating a patent. So you could only go to one company at the time, Myriad Genetics. And also people covered this news. And I thought, well, that just seems strange. If you want to get another second opinion for like dental work, you can go. If you want to get a second opinion for X-ray or your chest, you can go to another doctor. But for genetic sequencing, there was no place to go except for one company who owned the gene because they owned the gene. And so it was unavoidable for any modern day geneticist. If you just sequence a gene in your lab or by yourself, you would be infringing a patent. And, patent, just to remind you what a patent is, when you own something, a patent to even exist first has to be new and useful. This was established in 1790. It also has to be non-obvious, which was added in 1952. It's in the Constitution, so Congress shall have the power to promote progress of science and useful arts by securing for limited times to authors and inventors the exclusive right of their respective writings and discoveries. So it's encouraged by the Constitution itself to say we should encourage innovation, encourage patents, that people file patents. But it has to be, all of these things, new, useful, non-obvious. And we thought, well, it's not really, the genes existed for, at this point, millions and millions of years. I wouldn't say it's new. You didn't really invent the BRCA1 gene. It's definitely useful and non-obvious, a lot of the techniques. But we argue based on what's called this 101 claim that it's not new. And so the actual claims in the patents were language that looked like this, where any isolated DNA that Myriad would own that DNA and any variation of that cDNA and anything that had at least 15 nucleotides of either of these claims. So they would say anything that's the BRCA1 gene or anything even close to it, they own that gene. I thought, well, that seemed pretty non-specific. You've all been doing alignments and looking at bioinformatics tools. 15 nucleotides is very short. In fact, it's so short that literally every single gene has at least one 15 mer from BRCA1. So basically, we have this plot with analysis that every single gene in the genome has at least one 15 mer that's in BRCA1. So it's impossible. The patent just didn't even make any sense. We published this around the time of the case was in the spring and going through the courts. And it's not just matches in protein coding genes. Remember, the genome is 2% exons. The rest of the genome, we looked across all the rest of the genome. There's many pseudogenes. There's even a BRCA1 pseudogene. You can see here that's on one of the chromosomes nearby. And of course, BRCA1 is not just important for breast or ovarian cancer, but it has important for many cancers, like look at lung cancer, it could be prostate cancer, thyroid cancers. Many other cancers have mutations BRCA1 because it deals with DNA repair. So it's not surprising we'll see this mutated. So it's important for many other cancers because it has pleiotropy. It's one of the most important words for the course and for genetics. It's just that one gene can have many functions, which BRCA1 does for sure. So we filed a brief to challenge it. This is an example of one of the briefs to file it and challenge it in court. And we wrote op-eds in the Washington Post and said the Supreme Court should invalidate the patent on human DNA. And the CEO of Myriad Genetics said, these guys are morons and why are they writing this? And so we put up a big public battle. So I'll probably never get a job at Myriad Genetics, but that's okay because I think they shouldn't have owned the gene. They didn't own the gene. ACLU put up parts on their website about do not patent my genes, went all the way to the Supreme Court. This is us on the steps of the court and taking questions. Right after I saw Eric Lander and Francis Collins high five after we felt like it went well in 2013, challenged it in court, and then it was claimed to be rejected on June 13th. And basically the test for BRCA1 sequencing went from $4,400 to a few $100s in one day because we took away a patent because they didn't actually own that gene. I love patents. I have patents, but if you didn't invent something, you shouldn't have a patent on it, like the gene itself. Actually, the lawyer in the Supreme Court oral arguments, he said, the gene didn't exist until we found it. And I thought, I know trillions of human cells that would disagree with that statement because they've been using it for millions of years. But in case this was a battle, so if any of the legal challenges you'd like to think about, is we won that case. But every 2-3 years, there's new bills that get introduced to say maybe we should bring back the patents and different software and genetic patents. So far, we have deflected those attacks. But just my bit of personal history of like when you're in a moment in something, you think, well, that seems weird. Is that right? Maybe it's not right. And sometimes it will take 5 to 6 years of battling and litigation to get there. But we did, in this case, win in a sense. So trying to keep it to a half hour. And so, so could we just correct, say, the bad gene? So, for example, so I want to close, that's a bit of a battle legally, ethically, what's been done, the ones I've been involved, some of which I've been involved personally. But it begs this question of, okay, well, if you have mutations you don't want, you heard about genetic technologies, couldn't we just correct them and fix them? And then you wouldn't even have to worry about it at all because you could just fix your bad genes and have designer genomes. And this was first done, the very first example in a human embryo was done in 2015, that it was possible to do in human zygotes. This was done, perfected later that year. This was, more Chinese scientists showing that you could modify human embryos. This was done then also in pigs, where you could do multiplex genome editing and genome CRISPR for 60 genes at one time, which led to efforts in xenotransplantation. I'm not talking about that today, but it's another fascinating data set we have that's looking at xenotransplantation, where you take a gene, one species and move it to another, in this case, from pigs to humans. And I think, as most of you probably know, the real landmark moment happened when He Jiankui edited human embryos and actually deleted the CCR5 parts of the CCR5 gene to make the embryos resistant to HIV for parents that had HIV, that were HIV positive. He said, okay, I'll take CCR5 because that is, a superpower, right? So let's just delete that and make it so they don't have to worry about HIV. But the problem is, if you delete one gene, if you lost CCR5, you'll limit the ability to stop West Nile virus, because CCR5 actually in its functional form limits the viral loads during West Nile virus infection. So even though you're more susceptible to HIV when you have a CCR5 locus, you're less susceptible to West Nile. So it's a balancing act between different viruses, because it can save lives because it has pleiotropy. Because one gene, whenever you're editing one gene, you have to always think what else might be helping with positive or negative, what's called balancing selection. And so to that end, though, so that I want to put that as a point, but then also point out what's already happening. So, for example, in embryos, this is a picture of Sherkrat Metelipov, and I think he looks a little bit, he could smile there. He just cured like a disease gene. I think he should be smiling there personally, but he's not smiling there. But he fixed a disease gene in viable human embryos. And you can now do different methods of CRISPR, like prime editing, which doesn't involve the double-strand breaks. And this is great work by David Liu that's shown that we could probably select probably 90% of human disease genes we could probably fix with this method. And so, adult trials for sickle cell disease began in 2018. This was one of the very first sort of examples of using CRISPR in patients somatically. And the idea is, if you have, when you were a fetus, remember when you were an embryo, you were up here and there was Marvin Gaye and there was wine and candlelight. When you're a fetus, you have a different kind of hemoglobin that carries oxygen around, called fetal hemoglobin, but it goes away before you're born. You can see here in brown, eventually you get alpha and beta hemoglobin that carry all of your oxygen. But the simple idea of this clinical trial is, well, if there's a problem with these genes, we still have fetal hemoglobin inside of our genome. Why don't we just turn it back on? And that's, in fact, exactly what they did. This was the first pilot data in 2021. You can see they basically took here a Cas9 system and then deleted out this enhancer locus that's actually repressive element. that keeps the gene off, and they just got rid of it. So they made it so fetal hemoglobin in blue turns back on for these first two patients. And it was so successful that it's basically within 2 years it became an FDA-approved therapy. It's kind of extraordinary to think of a somatic CRISPR treatment that's now actually an FDA-approved therapy. That's extraordinary. So this is no longer hypothetical whatsoever. And one of the first recipients of this just last year actually climbed Mount Kilimanjaro, Jimmy Olagahiri, who was actually just met him a couple months ago at a conference. And he's walking around climbing mountains and now has actually no longer has sickle cell disease. And so and just actually a few weeks ago, there was another example of actually using somatic CRISPR to treat, in this case, carbonyl phosphate synthase deficiency, which builds up to a lot of basically a lot of ammonia in the blood, could be very toxic. In this case, is a baby that has basically so far looks like it's been cured by the disease that was found within a few weeks of being born. So really an extraordinary time in which we live. And in this case, for this NEJM paper, they showed that they did this CPS1 modification involved an adenine to inosine change, and then also another modification. So getting even base editing down to one specific base and getting the change happening, which is really extraordinary. So and the last of the kind of vignettes is that this is a great other example with Rudolph Janer that showed you can even change DNA methylations. You can have a DCAS9 system or modify other different areas of the genome by during adding or taking away methylation is another sort of feature that's been shown in the literature. So it's really a fascinating time to think about, well, maybe we're not just stuck with, this basic DNA. Should we upgrade our DNA or modify it? And the answer is yes, you can, and yes, we have, but you have to always keep in mind pleiotropy. If you change one gene, it might decrease the risk of one disease, but also increase the other. So just always keep that in mind. But to that end, though, there's an ongoing $200 million of a somatic genome editing program. They're now in phase 2, that should say at the bottom there. I just updated the slide this morning, but it is phase 2 right now, is ongoing to create these. If somatic genome editing were perfect, you wouldn't even have to do things as an embryo. You could somatically change all the cells in your body, which is not perfect yet, but if it were, this leads to a really different view of biology. I think that you could modify things long after they're differentiated. And to that end, we actually, just as a philosophical point at the end, we thought about what are protective loads that could help you in spaceflight in the future. And this is a kind of a fun table of what are all the things you could potentially modify that would help you survive on other planets, just as a fun vignette of some of the same genes showing up on a table that have some human and mouse data that could help you survive in faraway places potentially someday. And with that, I'm trying to keep it to half an hour. Last sort of perspective piece is there was a time that IVF babies and in vitro babies, test tube babies, were viewed very disparagingly and viewed that they were not like real people, that they didn't have a soul, that they were just outcast. But eventually, now 1% to 2% of babies are born by IVF, and it's very normalized and essential, and enables people to have liberty to have reproduction when they otherwise wouldn't. And Jennifer Doudna, Nobel laureate, says for CRISPR, she suspects that maybe CRISPR will someday be the same, hopefully, but we'll see if that happens. And with that, happy to take any questions. I know we're to the top of the half hour or the bottom of the half hour or at the end of the class, and happy to take questions, anyone online or in the room. See the chat. Okay. Any questions, thoughts, anyone? Yes.
So I know a big concern with the CRISPR editing is the off-target effects. I'm just curious what your opinion is on that, or what say the field thinks about it.
Great question. Yeah, and if people got to run, please go ahead. So that's actually before prime editing, I think before this happened, this was a really big concern about 10 years ago, even 5-6 years ago. But there's also other variations of prime editing that are even better, though, and David Liu published 5 years ago. So I think now we can reasonably say you'll be editing only 1 or 2 loci, maybe even just 1 loci with some of the newer prime editing methods. But there's actually a whole somatic editing consortium at NIST that's looking at this for off-target effects. So you have to be sure. But I think we're getting to the point where the number of edits that would be off-target would be less than just the somatic mutation rates. So then you could say, well, it's not even that different, just like living on Earth in terms of background mutation rate. We're close to that yet, but it's a great question, and it has to be. You have to check for sure. Yeah. So that was actually another critique from He-Jin Gui for his work. Is he used like the very early version of CRISPR that for sure had off-target effects that are awful, and so we don't even know. I mean, as far as we know, they're alive. We don't know what the impact was there. So that's the danger of taking a really bleeding-edge technology and then jumping into a clinical trials. Like, well, you could actually, with just a couple of years, it might be something dramatically better. That'd be much safer. But the other biggest problem. Actually, I encourage everyone to read this article and other ones. Is that the biggest critique? Actually, David Liu is the one who leveled it. Is that for what He-Jin Gui did, there was no unmet medical need. The biggest thing for any interventionist think is do I need to do this? And in this case, they had antiretroviral therapies. They had ways to keep HIV at bay if they were really worried about it. There was no reason you need to modify embryos to exist, on the world. There's plenty of ways for them to survive without this really risky procedure. So I think it's true for medicine and also most sciences. There's something, am I doing something that is an unmet need, in this case, a medical need, but even just in general, am I doing an experiment that will help reveal something either with positive or negative data? And if the answer is no, you maybe shouldn't do it, I think, especially medically in this case. So yeah, good question.
For these somatic mutations that you're like injecting into a person, don't they have to be mainly through like hematopoietic or like blood cells? Like you can't really like do it in like a muscle cell, right? And then have it like proliferate throughout the body.
Good question. So actually, a lot of on the website, the NIH website, a lot of the work, most of the grants now are on delivery mechanisms. So are they nanoparticles or the variations of lipid encapsulation that get you to the right part of the body? It's the hardest part of the therapy. It's been the hardest part of gene therapy since the 80s, is can you get a therapy to one specific type? If it's a retina, for example, or something you can inject directly into it, that's easier. But from a lot of the other targets, they're harder. If they're hematopoietic stem cells, it's a little bit easier, but even those can be hard. But I mean, I would argue that a lot of the challenges, Ben, we didn't know the epitopes in single cell profiles for every cell type as well as we have learned in the past, say, 5 years. The Human Cell Atlas and other projects finally gave us a really rich map of every epitope and combination thereof that lets you target one specific cell type, but we didn't have it. So for, I'd say not till 5-6 years ago did we have a good enough map. We had things like CD4 and CD8 would define T cells, and CD19 could be B cells, but there were a few markers, but not as many as we have now. So my optimism is that we'll have enough combinatorial markers that you get really good specificity for cells for delivery. We got to get the nanoparticle to the cell, but we didn't even know what to put on the outside of those particles until the last. Or we knew some markers, but not as many. So I'm hoping with a good combination, you get more specific. But they can't be toxic. They're going to go to the wrong place. They have to get cleared by the vasculature. There's other problems. Yeah, but that's, but at least we know what to target now, I'd say.